background, followed by Candice Gardner about the industry impact before the actual award is handed over by Dean Robinson. And then there will be a short answer, maybe not that short, from Sherret and Helen Chase in response to this award. And I also look very much forward to what you have to say from your experience and history here in Ames. So today, the CALS Award, College of Agriculture of Life Sciences Award, Sustained Excellence will be given to Sherret Chase. First, my name is Thomas Lüberstedt. I'm director of the R.F. Baker Center for Plant Breeding. I'm also founder of the Double Haploid facility. And it is for this reason a special honor, as he has been to be the first speaker in the ceremony for Dr. Sherret Chase, as he is the father of the double haploid or DH method in plant breeding. The most important discoveries were made while Dr. Chase was a faculty member here at Iowa State University. He was, however, hesitant to call him father of the DH method because of the limited role of the fathers in this process, as they only contribute to the, they do not contribute to the genomes of the double haploids. They are merely serving as inducers. And inducer of the DH method also did not sound right either. And for that reason, the subtitle is Origins of the Double Haploid Method. Dr. Chase's daughter, Helen, who is in the audience, along with a couple of family members, I think it's meanwhile 8 family members, supplied me with a large and impressive package of materials to document Sherret's life and accomplishments. Given limited time, I can only pick and choose. Dr. Chase received his BS, this one here, at Yale University in botany in 1939, so a long time ago, which was followed by a PhD in plant cytology at Cornell University. And it was quite different times. In between, he also served 3 years in World War II. He was in the combat, or was combat lead navigator, earning there the Distinguished Flying Cross. His PhD dissertation was on preliminary studies in the genus Nias, which are aquatic plants. In some years, this may actually come in handy for Iowa, I thought. He also conducted non-thesis pioneering research together with his advisors, Dr. Sharp and Randolph, on haploidy in corn, laying the foundation for his research at Iowa State University. Dr. Chase joined the Department of Botany and Plant Pathology of Iowa State in 1947 as assistant and later associate professor, before moving on in 1954 to decide ag research, joining the plant breeding industry. It is during his time at Iowa State that he did his groundbreaking research regarding the DH method, and then felt compelled to introduce it into practical plant breeding. He later rejoined the academics, first at Harvard, then for a long and ongoing period at SUNY, New York, where he's living. So what is it all about? Ultimately, the DH method helps to accelerate the development of inbred lines with this breeding process and genetic gains. It enables to obtain inbreds in 2 rather than 6 to 8 generations. This slide is also showing that Dr. Chase noticed that while surfing the internet probably, that there's a double haploid facility at Iowa State. This was in 2010 and led to 2 visits with his daughter Helen in 2011 and 2017. 2017, both together, were giving a seminar, and it was really memorable for everybody who was participating there. Back to the DH method. As mentioned, the male plants only serve as an inducer, as you can see on the slide. The The goal is to produce plants carrying only the maternal genome and subsequently to double it. It's a rare event, resulting in instantly genetically fixed lines. It is in this chain of events It's in this chain of events that is necessary to move from initial induction crosses to finished double taproot lines that Dr. Chase discovered and established a series of scientific firsts. He was the first to discover that the males, in fact, are important with regard to the frequency of haploids, and that did lead to the concept of haploid inducers. Another first is because haploids are rare in a cross, it is necessary to effectively identify those haploids. And for this, color markers were incorporated. This is another method that Dr. Chase was the first to develop. He was the first to develop methods to effectively double the genomes of sterile haploid plants, to turn them into fertile, and with this useful double taproot plants. He discovered that some genotypes have a tendency to spontaneously double in their genomes, which is valuable also. And he was the first to develop actually a DH line, including the first commercial line that was leading to commercial hybrid. However, both academia and industry can sometimes be slow to appreciate new methods. Returning to the presence. The list of firsts that Dr. Chase can claim is indeed quite a bit longer. That's what I wanted to say. And it shows how a visionary scientific concept requiring various obstacles to be overcome was successfully turned into an important breeding method, concept proven. So on the right side, you can see him standing in front of the first double taproot line. However, both academia and industry can sometimes be slow to appreciate new methods. And returning to the presence, in our own research, we were looking for genotypes with a tendency to double their genomes. A reviewer of a recent article submission commented that this is a hot topic, which I felt is ironic, given that first research by Dr. Chase was already done in the 1950s. With regard to commercial use, Dr. Chase stated the following at the Corn Industry Research Conference 1951. I hope you gentlemen will find my enthusiasm for the possibilities of the monocoid work justified. He didn't like the term haploid, by the way. And I was, by reading that, just picturing the audience and thought there was probably sitting a completely unenthusiastic audience, and not like you, and was really not ready to move on this method. So it did take a few additional decades actually before haploids in the DH methods became fully integrated into commercial breeding programs as they are today. This is something that Dr. Gardner will talk about in a few seconds. And personally, I deeply appreciate this foundational research that Dr. Chase has conducted many decades back here, which is still leading to exciting research opportunities and hot topics today.
Hello, now we're working. So I'm Candy Gardner. I'm a USDA ARS scientist here at the Plant Introduction Research Unit on campus. And Dr. Chase, it's an honor just to stand here and be talking about you. So we know that there's a whole string of activities that have to happen between basic research and its application and the realization of its value. And so there's a series of investigators that have contributed over a long period of time to where we are today in the use of haploid DH technology and now spontaneous doubling. And I listed a few of the people here whom I'm sure have consulted you many, many times, Dr. Chase. I can't imagine them not picking your brain at every opportunity. We didn't move forward. There we go. So we know that the adoption by commercial maize programs are widely spread now, and its adoption in public programs has been slower due to the availability of resources and the infrastructure in order to do this. It's hard to know exactly how many DH lines are generated in the private sector programs annually. We know it's several million. Because of their genetic uniformity, it's made genomic selection strategies feasible as well. It depends on accurate genotypic information. It depends on uniformity across loci. And so they've been able to rapidly deploy genomic selection strategies, not in any small part because of your foundational research. Here in Ames, Dr. Luberstadt has helped us generate many double haploid lines from exotic maize landraces that have been previously unsampled and previously unused in maize genetics. And so we've been able to provide maize genetic resources that are a research tool that weren't feasible before because they were so unadapted. So just for fun and trying to figure out how you go about measuring the impact of his work, I thought, well, let's look at the patent database and let's see what we find. So if you do an unrestricted search in the US patent database and you just put in the word haploid and you don't restrict it to any particular field, you get almost 15,000 hits, okay? And these things cover microbes, they cover plants, they cover fungi, all over the place. Then if you restrict it to haploid and maize and just mentioned in the abstract of the patents, you get over 11,000 hits. There's a lot of people that are using this technology. And this doesn't have anything to do with the number of DH lines that have been developed. This is just impact, direct impact, as evidenced by patents. If you search Chase and haploid, you get almost 2,000 hits. And that's because of their references to your work. If you look at embryo rescue and haploid, you get over 2,400 hits. The list goes on. And then you get into methods development. There's a 2019 patent for simultaneous gene editing and haploid induction. There's patents for highly transformable elite lines. Patents for spontaneous chromosome doubling. And then as a subset of that patent, automated systems that can help you tissue sample and identify those for advancing. It just goes on and on. So the impact you've had is just simply tremendous. So along with our newer technologies, we have questions and we have concerns. Oops. Thank you. We've moved to lots of short cycle time, multiple generations per year programs. And these aren't, the value from these programs have been enhanced by the use of DH technologies. But along with those come risks. As we go faster and faster and couple genomic selection with them, are we actually increasing or decreasing the genetic diversity used in these programs? We already have evidence from the dairy industry on the effect of genomic selection on diversity in dairy breeds. So there's real strong evidence that diversity quickly drops in a program without deliberate efforts to continually introduce genetic diversity. Well, that's a concern that we have to be aware of. With every technology, there comes new learning. So we have a lot of opportunities today that we did not have before. We can find map important traits. We can do it in better backgrounds. And coupled with other new technologies, such as gene editing, we can rapidly integrate genes and traits from non-adapted germplasm into elite backgrounds if we know something about sequence, gene function, etc. There are many crop communities that are very envious of the breeding systems that have been developed as a result of your research, Dr. Chase. So we'd like to express our gratitude for your vision, your insights, and your persistence. It took a lot of persistence, I'm sure, to develop this actionable understanding of biological complexity. So thank you. Someone needs to introduce you.
Hi, everybody. My name is Dan Robus, and I have the pleasure of being Sciences. Great pleasure for me to be here today with you. I know that we are in the presence of greatness, Dr. Chase. I give you this award to recognize your sustained excellence. I think all of us have had mentors and role models and moments in our lives when we're inspired to push forward and do more than we thought we could do. Ourselves with people who will inspire us. Sustained excellence can refer to both the longevity of your work, how long you do your work. I think in Dr. Chase we have both. Someone who lasted, and someone who has also lasted and good inspire us. So I'm inspired by Dr. Chase. Scientist that he has been and is, give yourself the opportunity to be inspired by others. Work that we just heard about, which was in the early 1950s, and Crick of the structured DNA. I knew that at the end of that short article in Science magazine was profound sentence, really. It was something that not escaped our attention, right? You remember this? You guys have all read that, right? I read it, and I'm not admitting it. Anybody in here not read that article? I don't admit it, if you're one of them. Anyway, the sentence in there was something like, it does not escape our attention that this structure might explain replication and heredity. That's because they allowed themselves the opportunity to think about the importance of their work. So as we become more and more engaged, deeper and deeper, Dr. Chase did early in his work, take the opportunity to step back from your work. Why is this important? How can others benefit from it? Sometimes we get too deep in. So I ask you to think about Dr. Chase and his work and let that inspire. I want to say something too, because we're here at a university, young people wanting to be in the next generation. I have a homework assignment for all of you who are students. I'm joking, don't you? I have a book I want you to read, please. Yeah. Serious? Take out your pencil, write this down. Great scientist named E.O. Wilson. Some of you may know of him. Harvard biologist. Worked in the realm of biodiversity. About 15 years ago, published a Pulitzer Prize-winning book called Letters to a Young Scientist. Letters to a Young Scientist by E.O. Buy it and read it. You can get it on Amazon probably for a great book to read, and it will inspire you. I think Dr. Chase read that book, you would find it well. And it's about looking into your science and for others. Find a role model and stick with that person. Dr. Chase has stuck with his work too. This idea to present an award to Dr. Chase, presented by Thomas and I as we walked across the quad about 6 months ago, about a very unfortunate topic. Let's talk about something that's much more interesting. Mentioned to me Dr. Chase and his work. So I have brought with me today 2 things to recognize this wonderful event. Certificate suitable for training, already present for you to recognize your sustained excellence. Another, which is an award that we typically give to alums of State University. Let's thank Dr. Chase.
I brought with me a version of me because she has good hearing. I do not. And I really would agree with her since she will represent me in voice. Helen?
So we're standing here together. And thank you very much to the Iowa State community and the College of Agriculture and Life Sciences. Thank you very much. I feel honored and I truly appreciate this award from the College of Agriculture and Life Sciences and Iowa State University. This is a rare gift for an old man. My family, my wife, Catherine Compton Chase, or Kenny, and 2 daughters, and I moved to Ames and to Iowa State University in February of 1947. At about the same time of the year. Our daughter Cece was 1.5 years, and Helen was only 6 weeks old, having been born in Ithaca just at the time I received my doctorate from Cornell. My thesis research at Cornell related to the water nymphs, the naiads, the genus Nias, aquatic flowering plants that present their total life cycle underwater. I studied the 7 known species to determine their chromosome differences. Just prior to my entering Cornell, the botany department had a number of superlative graduate students and professors of genetics. Barbara McClintock received a Nobel Prize for her work on jumping genes. She really ought to have received the Nobel Prize for her work in determining that genes were part of the chromosomes. This history defined the department. The relatively new field of genetics was very dynamic and was humming at Cornell. I encountered 2 pivotal experiences in my progress through my coursework. My 2 primary professors, Lester Sharp and Lowell Randolph, were important influences. Sharp's course on chromosome biology and haploids and parthenogenesis, and Randolph's work on corn, maize, cytogenetics, particularly his interest in the occurrence of haploid plants and the possibility of obtaining homozygous diploids directly from haploids. When I arrived at Ames, I had come prepared for a very busy spring planting season. With the help of Sharp and Randolph, I had started a planting the year prior, where in May 1946, Randolph assembled genetic stocks and promising source materials for developing commercial inbreds and planted a special nursery of promising dent and sweet corn stocks. When the pollination season came on in 1946, I increased the stocks needed and made a vast series of marker crosses. Randolph had provided me with some unusual stock that I could use as a male parent. It was one that Randolph referred to as, and I continued to call it that also, brown ligulus. What I did at Cornell was to develop the ideas and to get the first work started on testing those ideas. Once at Ames, I tested those ideas successfully. I received excellent support in my move here to Iowa State. Dr. Sprague, the leading USDA corn geneticist and corn breeder in the United States, stationed here at Ames, provided planting space with his own corn nurseries on his own private land, pollination supplies, seed stocks, and most importantly, the Iowa stiff stock synthetic that he had created, and more. Most of my firsts in developing the doubled haploid method was accomplished here at Iowa State. Most of my publishing occurred here during my Ames. I also learned a great deal about how to get a successful nursery. My first planting during the spring of 1947 was a total washout. Over Memorial Day weekend, it rained, and then it snowed, and then it started to sleet, and then rain again, and more snow. My second planting created a very successful nursery. My work went very well, and I published. But I made a couple of publication errors. Errors in the sense that I realized at the time how noteworthy, how newsworthy some of my work was. For example, I was able to get the first seed by self-pollinating a haploid plant. That event really deserved a major article in a major publication. I knew that this was all very much to be expected by the people close to the cutting edge of research, but not quite ho-hum. And I thought it was just a little item of interest that I had been able to get some seed. Looking back in terms of building one's own professional reputation, I missed a major opportunity here. I made another publication mistake. That is, I objected to the word haploid because it was very much misused. It was kind of an interesting situation. I began using the term monoploid. The haploid term is paired with diploid, as in haploid and diploid. Then another kind of system of naming things developed. They took the words di and ploid as a fold, as in two-fold parts. So you diploid, a triploid, a tetraploid, and on up and down the line where you will find a monoploid. My haploids in corn were clearly monoploid. They had only one set of chromosomes per cell. The word haploid was often applied to any parthenogenetic individual. If that individual were a tetraploid, the haploid, so-called, was actually, in terms of genomes, a diploid. So I used the term monoploid, but monoploid did not catch on. It is a word that would not have been picked up in a citing system as often as the word haploid. I succumbed. Through a friend of my wife, Kenny, Mr. Brown, who was in charge of the farm at the experiment station, Mr. Brown and his wife were able to find a little house on the edge of campus on Ross Road that they thought would be a good starting house for a growing family. I was able to walk to work each day. The alternative was to live in an overnight pop-up development of Quonset huts called Hamel Court. Kenny and I expanded our family with 3 more children born in Ames, and our usable space in the house expanded into the attic and into the basement. My daughter Alice was number 5, born in 1953, just before we left Ames for DeKalb County, Illinois. Alice is here with her husband and Alice's son, Christopher, my older sister, Cece, and her daughter, Tisha, and Tisha's son, Chris Harrison, are also here. Some of the men on the staff that I worked with or that helped me included Dr. Lush, who was a world-famous animal geneticist. He complimented me by sending all of his graduate students to take my course in cytogenetics. Dr. Sprague was the principal corn breeder in the United States. In my department, Dr. Sass was a very fine plant anatomist and very helpful to me in setting up techniques for doing microscopic studies. Ames had a very fine statistical department. A young professor there, Dr. Kempthorne, was my key contact. It is interesting that Henry Wallace had started 2 things at Iowa State that were later very important. One was to set up a statistical lab. He had found that the U.S. Department of Agriculture did not know how to analyze its data, its numerical data, and encouraged the formation of a statistical lab. I think it was this was the first such university lab in the country. It spawned one of the most important statistical labs that exists today, the one at the University of North Carolina. Dr. Kempthorne helped me present probability data for occurrences that were very rare. That was kind of a special twist to how one should look at such data. Dr. Lindstrom was the head of the genetics department. He was very helpful to me by making available to me the greenhouses that were under the supervision of the genetics department. My office was on the 4th floor of the same building where the state seed testing laboratory was located. They had germination chambers, kind of like big refrigerators, and there were trays in them, maybe 10 trays to a refrigerator. They used a special kind of blotting paper and put the seed on the blotting paper and covered it up with some other sort of paper, and then would run a germination test to see how many soybeans or peas or something else out of 100 would germinate. I realized that this would make my work so much easier if I planted my marked seed on the germination trays, and then, without any digging up of the seedlings or damaging them, I could locate my putative monoploids. That was a big, big step forward. I remember an event when my 3rd child, a son, was visiting with his mother, and he, in his diapers, plopped right down on a seed tray, sacrificing about 300 root tips. By the way, my office had been the office occupied by George Washington Carver, and I had use of his stand-up working desk. My 2 oldest daughters remember particularly this special office and the desk and the banana tree in the greenhouse. Kenny and I had a wonderfully active life in Ames. I became a member of the local Lions Club. I was the faculty advisor for the fencing club. When the new Edwards Elementary School was opened, I was the first president of the Parent-Teacher Association. The PTA was full of professors. It was quite an intellectual group. Kenny was involved with the League of Women Voters and had her own play group for our own children, plus several neighbors' children. Kenny had a remarkable compliment paid to her when we announced that we were leaving Ames for Illinois. People started to invite us over for dinner. And for 45 days, it's hard to believe this, 45 days, at least one meal a day, we ate somewhere else as a guest of someone. And the last week, We were even eating breakfast as guests elsewhere. However, the very last meal that we had in our house, and it was very simple, was cornflakes. And we sat outside on the stoop and we had a guest. That guest was Henry Wallace. Our last dinner party was celebrated on November 27th, our 10th wedding anniversary. The guests were all our very closest friends. Some of these names stuck around here at Iowa State for a number of years, and many of them may be still recognizable. They included Allison and Howard Hamilton, Nikki and Bill Buck. Nikki was Dr. Lindstrom's daughter. Ruth and Joe Foster. Rhea and Sid Fox. Janet and Jack Wallen. Barbara and Bill Owens. Kathleen and Joe Keller. Ursula and Fred Smith. Tilda and Fritz Schlank. Rose and Paul Sharp. Dan and Susie Zaffarano. Franny and Dick Powers had already moved to Cold Spring Harbor on Long Island, and Betty and Jim Coles were away. These were our very wonderful bunch of friends. The Zaffaranos purchased our little house when we moved. Kenny and I had 5 children. The Zaffaranos grew to 6 children, and that little house grew along with them. There has been much through the years to bring us back to Ames. We loved living here. We had deep friendships, which we have kept up with. Not many have lived as long as I. I have been blessed with good genes and good nutrition. My mother was a master horticulturist from the Ambler School in Pennsylvania, and my father was a civil engineer responsible for overseeing the building of many bridges across the United States. Since we are in Iowa, one of those bridges is the one over the Mississippi, the Rock Island Bridge between Bettendorf, Iowa, and Moline, Illinois. There are 3 bridges. We tried to cross it when we were coming up, and one is Route 80, one is Route 280, and right in the middle is 74, or 76, I guess it was. That one was closed, but that was my grandfather's, and the one that was Route 80. Other Mississippi bridges included the one at St. Louis and the Huey Long Bridge in New Orleans. Thank you very much to Dean Robeson and to Professor Luverstadt for this award. And Candy, thank you for your part in this too. Receiving this from the College of Agriculture and Life Sciences at Iowa State University means a great deal to me. I would like to learn so much more about the exciting work being done here at the doubled haploid facility. I have roots here, both parthenogenetic, my haploids, and heterozygous, my family. Thank you very much.